In this mini lecture, we're going to talk about transposons, also known as mobile genetic element. These were discovered by Dr. Barbara McClintock in the early 1940s. She noticed that there was a difference in pigmentation between maize kernels. And this difference in pigmentation was also linked with a huge difference in terms of genetic composition of both type of kernels. And later on, she came to realize that this change in pigmentation were actually associated with a chromosome breakage that were happening at the same locus in various plants of the same strain. And eventually, what she noticed as well as she continued researching is that those breakage could actually jump or move it to a different part of the chromosome as well. And what she did, she renamed such mobile hotspot for breakage in the chromosome and gave it a name that is now known as dissociation element. And this dissociation element, they did not also operate alone. They needed another element that was referred to as an activator. And when the activator is present, the change in the dissociation element are actually unstable. As you can see in the picture that I have somewhere here in the note, what you notice is that you have a gene and there's this transposable element that comes and jumps in between that gene, and all of a sudden there's a change in the expression. And later on, when that transposable element jumps and move, there's also another change of expression. So what you see, those blue and white dots represent a kernel. So whenever there was a jump or the presence of the transposable element in the gene, you see that the kernel looked white, and as soon as the transposable element actually left the gene, the kernel looked now dark or pigmented, because the gene that she was actually interested in, or she was actually looking at there, code for a pigment which is known as anthocyanin. But when it comes to transposons, what we also know about them is that they do require the presence of an enzyme known as transposase. So there's a difference, or there are different type of transposons. Some of them can be autonomous and others can be non-autonomous. The transposons that are autonomous are transposons that are capable of encoding their own transposase, which is going to help in jumping from one location to the other. While the non-autonomous transposon, as you see in the note here, they rely on transposase activity of another transposon element. So next we're going to look at the different way in which this transposon jumps, which is going to be the copy and paste and the cut and paste mechanism.